has the for the whole day, so feel free to stop him and ask him questions whenever you and interact or to know the environment in his institution. But it is a pleasure for me to introduce today the speaker for our Wallace Coulter seminar series. I know Dr. Shmuel for, I think, maybe 15 years. He was one of the persons that I started following when I fell in love with fMRI and with the potential of that technique. She started working with Dr. Greenbaum in understanding how brain vasculature work, I think using intrinsic signal, and it's back in Israel. And then at some point, in 2005, I guess, we both applied to the same position at McGill. We were the two finalists. But I didn't coincide with him at the time of the interview. That position took a long time to open because of McGill procedure, and the Hope University where I had a very healthy counter offer. So I dropped that application and I moved to, and of course immediately, or a year or a few months later, I hear that that they got Dr. Shmuel, which is was at that time one of the top researchers in the area of fMRI and understanding how neurons and vascular work and communicate. Since then, so he first was at Tübingen, Max Planck, for 2 or 3 years, or 5 years. But also working with Nicholas Logothetti, but also was at the University of Minnesota with Camilo Ulke at some point. So he has been international and with the top researchers in the area. So his expertise is multidisciplinary. He has a very well understanding of the very multinational community and with diversity. And he has been very successful there in market. He was able to secure, I know that all this thing is here, but a very large grant from the Canadian government agencies in the order of $20 million. That's what I hear, like 5 years ago, something like that. He was able to buy the first high field MRI for monkeys.
Well, it's humans as well.
It's humans as well. But when I visited him, I think back in 2018 or something like that, he already was having those chairs that MRI compatible to put inside the scanner to scan monkeys doing certain tasks. That was, for me, amazing. And a previous visit, when he was establishing at that time, he was doing a lot of experiments with rodents. So he even has experiencing multi-species, from rodents to monkeys to humans. So I hope you enjoy today's presentation, which is going to be the foundation of how we can resolve a cortical column using technique that suffers some spatial resolution and temporal resolution like MRI. So I invite you to join me to welcome
back?
Yes.
Thank you very much. Thank you, Jorge, for this very generous introduction. It's really a pleasure to be here. That's my second visit to Florida International University, and I'm really enjoying it, and I'm looking forward to discussing with you. I think that the material I'm going to present is not easy, especially for people who are not in the field of imaging and high-resolution fMRI. So please do not hesitate to ask me. Raise your hand, ask me, and as Jorge said, I'm happy to interact later on. I'm here the whole day. So I'll be speaking about analysis of the spatial specificity of BOLD. BOLD is blood oxygenation level dependent functional MRI, and the mechanism underlying decoding information conveyed by cortical columns. So there is a long outline here, but I'll skip it. While I speak, I'll tell you, we completed one part, we go to the other, etc. So first, I would like to tell you about orientation columns and ocular dominance columns, neurophysiology and optical imaging. So on the left are pictures of David Ubel and Torsten Wiesel. They secured the Nobel Prize in the 70s based on the experiments in cats and monkeys, neurophysiology in cats and monkeys. Here's the experiment, or a scheme of the experiment: an anesthetized cat with eyes open, viewing a screen where stimuli are presented. And that's the way they presented stimuli at the time. So they had a projector, in this case showing a white bar. They were able to change the orientation of the bar and to move it from one place to the other. So as you probably know, neurophysiology is performed by having an electrode close to a cell body. So here is a scheme of cell bodies of 3 neurons, pyramidal neurons. The electrode is close to the cell body, and because of that, it can detect currents that flow outside of the cell, but these currents actually reflect the currents inside the cell. These are currents that nullify the currents in the cell, right? According to Kirchhoff equations and laws, when there are charges moving from one place in the circuit to another, there has to be a nullifying current going outside or going in a different way in order to balance the change in charges, right? Charges do not disappear. They just move from one place to the other. So the electrode is not really in the cell, but it can detect what the cell does. And with that, they can detect action potentials. So let's see what we detect or what Torsten Wiesel and David Ubel detected. When they presented horizontal orientation, they obtained, during the presentation of the stimulus in dark gray, only one action potential. The response is not very high. When they presented an oblique orientation, they were able to record 4 action potentials. So the response is just a little bit higher. But for this specific set, when they presented a vertical orientation, a bar oriented vertically, they recorded many action potentials. So this is called orientation preference. This neuron prefers vertical orientation. It responds a lot to vertical orientation and just a little bit to horizontal orientation. Now, we pursue such experiments with fMRI and with optical imaging. So let me tell you a little bit about optical imaging. This is the brain, the cerebral cortex of an animal model. The cortex is exposed after removing the skull and the dura mater. So what you see here in gray is actually cortex, the tissue, and in black are the pial blood vessels on the surface of the cortex. And we shine, in this case, 605 nanometer light, orange light, and we record video during the presentation of stimuli. We record video onto the computer. Now, why 605, or in this experiment 660 nanometer, so it's an orange light, and 530 nanometer green light are important for us. What we see here is the absorption spectrum of cortex as a function of wavelengths. So at 616, you can see that the deoxyhemoglobin in blue absorbs much more than oxyhemoglobin. please note that the scale here is logarithmic. The oxyhemoglobin in blue absorbs much more than oxyhemoglobin at 616. At 530, they absorb the same amount of light. So we can use 616 to estimate blood oxygenation, the difference between oxy and deoxyhemoglobin. And with 530, we can estimate changes in total hemoglobin, which is approximately representing cerebral blood volume functional MRI. Okay, so with optical imaging, I want to emphasize that the measurement is based on absorption of light, but the physiology of the response is identical to the physiology of the functional MRI response. The only difference is the principle of measurement. With optical imaging, the measurement is based on absorption of light. With functional MRI, the signal depends on blood oxygenation, on the magnetic properties of oxy and deoxyhemoglobin. But physiologically, it is the same. Now, I presented to you the results of Hubel and Wiesel with an electrode, changing the orientation. We can do that with optical imaging as well. We can present 8 different bars of 8 different orientations, as you can see here. Now, they produce in each pixel what we call a tuning curve. In this case, that's the response from one pixel. It prefers vertical orientation. We get the highest response in vertical orientation. And then you can see that there is a reduction in responses going to oblique, the right oblique, the left oblique, and almost no response for a horizontal orientation. Okay, so this is the tuning curve. Now we can do that for all the voxels in the image. Look at the scale bar here, it's 1 millimeter. These are cortical columns, and they are imaged from top, from the surface of cortex. Now the color, the underlying color represents the dominant orientation. So if we look at red here, red represents regions that prefer horizontal orientation. Cyan, which is here, represent pixels or regions that prefer vertical orientation. We do that by vectorial summation of the responses to orientation, pixel by pixel. Okay, so we can get the map of orientation preference. Hubel and Wiesel obtain results from one electrode, and we can now do the map. Now, let us do the exercise of including here 6 classes where we will insert an electrode. And I want to explain why we call these orientation columns. So on top, remember we marked 6 classes here. During the experiment, the animal is still in the experiment. And on top, we see the preferred orientation from these 6 insertions of electrodes in the very pixel where we inserted the electrodes. Now, at the bottom, we see as a function of cortical depth, from 0 to 2 millimeter, the responses of action potentials obtained also with presenting the same grating, the same oriented grating. So you can see that the response that we get with optical imaging, in this case, the preferred orientation is the right oblique, matches the action potentials throughout the cortical depth. We call these cortical columns because the entire column, the entire cortical depth, responds similarly to orientation stimuli in this case. And of course, you can see it also in the other insertion of the element. So these are cortical columns for preferred orientation. Now, there are also cortical columns for ocular dominance. What does the neuron prefers? stimuli from the left eye or the right eye. So ocular dominance columns and map, in this case, in human. First, how are they generated? And this is known from monkey research. There is input from the left eye to V1, area V1, the primary visual cortex. And there is input also from the right eye. And this input is segregated. It doesn't mix. It goes to a spatially complementary regions. And it creates a column, again, as a function of cortical depth and cortical area, 1 to 6. It creates preference to responses from the left eye or preference to responses from the right eye. Now, these were detected by optical imaging in monkeys, but they exist also in humans. On the upper side here, you can see a post-mortem flattened brain, flattened cortex. And in area V1, you can see a stripy pattern. So this is a patient that contributed his brain for research. And this patient lost one eye while he was already adult. The staining of cortex here is for cytochrome oxidase that labels oxidative metabolism. The eye that was healthy brought input to V1, and that's why we get the brown, the stained stripes. The eye that was unhealthy, he couldn't see with his eye, did not give input to V1. And in the areas that represent the input from the unhealthy eye, we can see bright regions that do not pursue any oxidative metabolism. These regions are not metabolically active. And you can see that the pattern is striping. There are stripes. We can digitize these stripes in V1 and look at the scale. So that's a magnification. It's 1 millimeter. In humans, ocular dominance columns are about 1 millimeter wide. So we talked, or I presented orientation column and ocular dominance column, and I move on now to functional MRI-based decoding in humans. It is also called in the literature multivariate pattern analysis. The idea is that with functional MRI, these algorithms do not analyze voxel by voxels. They take the pattern and they look at the multivariate pattern response. So here is a study that made decoding quite famous. fMRI-based decoding of information conveyed by cortical columns in human subjects at 3 Tesla. So what is the stimulus? The stimulus are oriented gratings, just like Shmuel and we presented, and just like we present in my lab to animal models. So you see here horizontal gratings and right oblique and vertical and left oblique. And next to each logo or scheme of a stimulus, you can see the tuning curve in polar coordinates. So in a horizontal, and next to the horizontal gratings, you can see the results of decoding. What do I mean by decoding? The algorithm takes all the responses and tries to predict, to guess what was the stimulus. Okay? So you can see that the algorithm here predicted very well that the stimulus was horizontal. Here, right oblique, very, very well. Here is perfectly right oblique and very, very well. Vertical, where the tuning curve is not that narrow, but still, it's definitely statistically significant. Left oblique, etc., etc. So, multivariate pattern analysis, coding, machine learning, these are all similar synonyms, can decode the orientation of stimuli presented to human subject 3 Tesla. Now this is really surprising. Why? This is surprising because these studies use boxes that are 3 by 3 millimeters, okay? 3 millimeters by 3 by 3. And as I already presented with optical imaging, orientation columns are really represented in fine scale. So this is just a scheme to show what do you see in a 3 millimeter voxel. You really see a mixture of very many orientations. When you see a mixture, how can you get even information from that, right? So this is surprising. This is just to formalize the surprise. In humans, based on the Horton result of cytochrome oxidase and ocular dominance, we expect that the cycle of orientation, one cycle going from horizontal to vertical and back to horizontal, is about 2 millimeters, one cycle. So how can we decode information with a representation of one cycle per 2 millimeter with 3 by 3 by 3 millimeter voxels? It refutes the Nyquist theorem, right? We don't have the resolution to actually get information with that. So that's the topic of the first part of the talk. And there were mechanisms proposed. So this triggered quite an interest. And let me tell you that Kamitani presented the decoding paper in 2005. The discussion is still ongoing. Like, people are still debating what is the mechanism. So what was proposed as the mechanism? The first proposal was aliasing of high spatial frequencies. So suppose this is the real frequency of the organization. If we use larger voxel, we get aliasing, meaning we get something that represents the organization, but at lower frequencies. Whoever studied digital signal processing is probably familiar with the term Nyquist theorem and aliasing. If we don't have the resolution to resolve the real frequency, we get something different, which is at lower frequency. So that was one mechanism. The other mechanism suggested fine-scale irregularities, which means in each voxel of 3 by 3 by 3 millimeter, there is a distribution of preferred orientation, but there will be just a little bit of bias to certain orientations relative to others. So here I represent the histograms of the preferred orientation in 9 voxels. And this little bit of bias, when you use the information from a pattern, not from a single voxel, you can decode it. That was the second mechanism proposed. The third mechanism proposed that in addition to the high frequency organization, to the fine scale organization of orientation, there are probably low spatial frequencies responses to orientation. So yes, there is the fine scale, the high frequency, but they ride on some low frequency responses that can be detected even by 3 by 3 by 3 millimeter voxels. So one of them is so-called the radial bias, where the orientation, or when we present vertical orientation to a subject, we get higher responses along the vertical meridian, along the visual field, which represents the vertical orientation, the middle of the visual field. So this is another possibility. So this is the third mechanism. The fourth mechanism, which I presented back in 2010, is selective responses of macroscopic vessels. I present here an ocular dominance map that I obtained at the University of Minnesota at 7 Tesla. So left eye is represented by blue responses, right eye with red responses. And here I represent veins. And I'm not going to get to the details. You can ask me later, how do I know that these were veins? But there are big regions on these veins that respond preferentially in a statistically significant manner to certain orientation. Now they can be detected, these are large veins, so they can be detected with 3 by 3 by 3 millimeter voxels at 3 Tesla. And a variation of that was proposed by previous Coulter, who suggested complex spatial temporal filtering by fMRI voxels. So he said, yes, the blood vessels, the capillaries, represent orientation very well. They represent the fine scale. But they drain the blood into venules and into veins, and there is some residual orientation selective responses in the large veins. So there is like a complex spatiotemporal filtering, which means that the veins can take the high frequencies and transform them to low frequency, and they still carry some information on the preferred orientation. So here are the 5 proposed mechanisms: aliasing, irregularities, low spatial frequencies, macroscopic vessels, and spatiotemporal responses that represent complex filtering. So the aim of this talk is to unravel the mechanisms underlying hemodynamic response-based decoding of oriented grating stimuli using coarse voxel sampling, 3 by 3 by 3 millimeter. Motivation, why is that important? Unraveling the mechanism underlying decoding of fine-scale organization. If we understand the mechanism, we can use the same method to search in an informative manner such organization in relatively unknown cortical regions. V1 is very well known from the monkey, but there are very many other areas in parietal and frontal cortex that we don't really know what they do and how they are organized. So if we understand the mechanism in V1, we can apply the same methods to detect organization, fine-scale organization in frontal cortex. So next I will talk to you about or present mechanism underlying decoding of the stimulated eye through a model. My student, Dennis Chaimov, pursued a model in order to, he created a model in order to understand the mechanism. Now, with this model, we address the question of aliasing, is that possible? And irregularities, is that possible? We didn't address the 3 other methods, the 3 other mechanisms. So what does the model do? It takes white noise image, and it filters it, low pass filtering with anisotropic kernel, with elongated kernel. What this creates, and of course there are parameters that need to be tuned in order to make the pattern similar to that of human ocular dominance. So we get the pattern, and then we simulate response to the left eye on the left and to the right eye on the right. And then we convolve with the point spread function of the hemodynamic response. Functional MRI is not point-like. You give a stimulus, it creates a spread of the responses, right? There is a response in one position, but there is a spread of the responses around it because of the draining of the blood to venules and veins. So we convolve with the hemodynamic response function, and then we simulate functional MRI sampling. Functional MRI doesn't sample in the image space. Functional MRI samples in the frequency space. So we simulated that, and you'll see soon that it's a little bit different. It creates different concept than what we're used to in image processing. Right, so we simulated ocular dominance in humans, and eventually we get voxel functional MRI responses. So the first question, is it possible that aliasing contributes to decoding of, in this case, ocular dominance codes? And the answer is not. And the reason is that because functional MRI encodes the information, gets the data in the frequency space, in the image space, we get sync-like voxels in the blue curve. We usually think about images as if each pixel represents a rectangular region with a certain intensity. But with functional MRI in the image space, each pixel of voxel represents actually a sync-based function, right? Now, if we look at this sync-based function in the frequency space on the right, and we look at spatial frequency that it can get information from, we see that the blue vector, the sync-based function, only gets information from very low frequencies. And this information has very low amplitude. So I show here the frequency space, the spectrum. Now, we superimpose the spectrum of ocular dominance columns, right? So the blue region, where the sync-based voxels can get information from, gets really very low energy that overlaps with the spectrum of ocular dominance. In contrast, if we were to use a box like pixels, the cyan pixels, we would get some information at higher frequencies. And so possibly we could decode part of the information with box like pixels in cyan. But functional MRI works with sync-based boxes, and therefore the level of energy that it can decode with fine scale organization is really low. So we ruled out the possibility of aliasing as a mechanism for decoding. So here are the conclusions from the model. Sub-boxes supra-Nyquist frequencies cannot contribute to the differential contrast, meaning we cannot do, aliasing cannot contribute. I don't represent here in detail how we got that, but the differential functional contrast shows only low amplitude contributions from irregularities of the cortical pattern. Okay, so we also ruled out irregularity, and for the sake of time, I'm not getting into details. And therefore, we concluded that we expect that functional MRI-based decoding in V1 relies in part on signal contributions of large-scale, non-columnar functional organization, right? So we tested aliasing and irregularities. We ruled out aliasing. We say irregularities may be a small part of the decoding, just a little bit of information, but we didn't test here low spatial frequencies, macroscopic vessels, and spatial temporal responses. Any questions up to here? Yes, please.
I wanted to ask, what is the technique you used to get the signals? Was it an optogenic inhibition, activation, maybe calcium fluorescence imaging?
All right, so you're talking about how do we measure functional MRI? The normal way, or the most common way to get functional MRI is to get the subject into an MRI scanner. Okay, MRI scanner, I think you have it here at the Florida International University, right? So it's an MRI scanner, and present, in this case, just visual stimuli. You can present somatosensory stimuli. You can ask the subject to solve mathematical problems and check which areas are active. In our case, we talk about presenting visual stimuli. Now, what this causes is a response, a hemodynamic response. Areas that are active, where the neurons are active, draw more blood. There is more blood flow into these areas. And this blood flow changes. Let me just say that the scanner can detect these changes. The scanner knows which areas change their blood flow, okay? Or the blood oxygenation. So that's the measurement we are talking about. Now, I presented optical imaging, which is a method we use in the lab in order to mimic functional MRI. It is also based on blood oxygenation and cerebral blood volume. The only difference is that in the lab, the measurement with optical imaging is based on absorption spectra of oxy and deoxyhemoglobin. And in functional MRI, the principle of measurement is based on the magnetic properties. Deoxyhemoglobin is paramagnetic, it's a weak magnet, oxyhemoglobin is very weak magnet. It is diamagnetic. So that's the measurement. The measurement of functional MRI is based on hemodynamic responses to regions where neurons are active. There is more blood flowing into it. Okay? Please.
You, aside from oxy and deoxyhemoglobin, like in the brain, do you consider like other components in the brain that could also have that similar magnetic, like for like maybe like cytochrome or something like that, that also impacts brain function, but similar magnetic? Is that something that was considered?
Well, so the brain has regions, but there is a variation of iron deposited in the brain. So for example, the basal ganglia is a region where there is a lot of iron. And we can detect it in both functional MRI and in structural MRI. But everything here, and everything in functional MRI is based on differences, on relative responses. So you show a stimulus, and you look at the percent change relative to no stimulus. So the iron, which is static, it doesn't change, is washed out. It's the same in the two functional MRI measurements. Okay? So functional MRI is based on hemodynamic responses. Mainly, 99.9% of the studies are based on blood oxygenation, meaning they measure the content of the oxyhemoglobin in the voxel, and 0.01 are based on cerebral blood volume. They measure blood volume. Okay? Thank you. So next, I will talk about mechanism underlying decoding of orientation stimuli based on measurement in CAT area 17 using myon cerebral blood volume. So myon is a contrast agent. We can inject serum, nanoparticles of serum, into the systemic blood. It is distributed in the systemic blood. And it creates much stronger functional MRI responses, much higher signal to noise ratio. So it's like, unlike the oxyhemoglobin, which is paramagnetic, a weak magnet, the serum is a very strong magnet. So we get much higher contrast to noise ratio with myon. So let's see how it works. In this study, well, let me tell you our conclusion. Fine scale irregularities do not contribute, and contributions from macroscopic vessels are not necessary. They are there, but they are not necessary for functional MRI-based decoding of oriented grating stimuli in CAT area 18. So aliasing, we already ruled out with the model. What we're going to test now are contributions from irregularities, fine-scale representation, and low spatial frequencies. And we actually say that based on the study, macroscopic vessels can contribute, but they are not necessary, right? The responses are redundant based on macroscopic vessels. So let's see how we do that. We present oriented gratings again to a cat, anesthetized cat, just like human and wizard. Only in this case, we use functional MRI of hemodynamic responses. We look at blood flowing into pixels or voxels that change the responses to the oriented grating. Now, in this case, we use a so-called phase encoding stimulation, but that's not critical to understand. Here is one image of functional MRI from area 18, and this is the preferred orientation. Remember, we presented that in color, and you can see the preferred orientation from minus 90 degrees all the way to plus 90 degrees. Whenever a stimulus in a voxel appears to be the preferred orientation, this voxel represents or responds in a high response, a functional MRI response. This is a time course, right? And the stimuli go around, so the orientation changes, and whenever the voxel whenever the orientation matches the preferred orientation of this single voxel, it responds at high amplitude. And we said we're going to test irregularities and those spatial frequencies. So for this, what I want to introduce is the measurement of cortical frequency specific contrast to noise ratio. So here's the response that we get for a specific orientation. And this is broadband, so we don't filter a specific frequency. Here's the noise, the standard deviation. We present to the cat the orientation many times. So we have also standard deviation. Here we see the contrast to noise ratio, meaning the mean response when presenting this orientation divided by the noise, by the standard deviation, we get the contrast to noise ratio. Now we can do the exercise of using a specific frequency. And the specific frequency is that we expected to be the frequency of the organization in cat area 17. And you can see that the organization now is more regular. So that would be the mean response or specific frequency or specific frequency of cortical organization. And then the noise associated with this frequency and the contrast to noise ratio. You can see that we get a little bit higher responses here and everything is more regular. Now, we can do that and we can present the cortical frequency specific contrast according to frequency as a function of different frequencies, okay? And this is from all voxels, but aligned to the preferred orientation of the voxels. So here are the results from two animals. So you can see, and we fitted here a Gaussian, you can see bimodal distribution. So we get the peak at the preferred orientation and at the preferred frequency of the organization, but we also get low frequency organization in these two animals. And this is the response of the cortical frequency specific contrast to noise ratio averaged over all the animals in the study. So again, we get the peak at about 1 cycle per millimeter, but we also get low frequency. We get a bimodal distribution. Now, what are the peak and the low frequency represent? The peak is the frequency of the organization. It is at the frequency of the organization, meaning, in this case, it's really representing this map, meaning the map where we know cortical columns are organized. I told you that in human, it's 1 millimeter per ocular dominance column. In cat, it's a little bit different. So that's what the peak shows. And we also get low frequency, right? Now, we did the experiments in cats, but we want really to learn about what is going on in the human visual cortex. The organizations have different frequencies of the organization, right? In humans and in cats. So we made an estimation of what would be the voxel in human that can detect irregularities based on what we see in the cat. In the cat, we see that the preferred orientation, the peak, is about 1 cycle per millimeter, but we get a lower response around this peak frequency that represents the irregularity. So what would be the voxel with which we can still get some information on irregularity? So this voxel in human would be 1.5 millimeter. And below that, there are the low frequencies in the cat. So there is a trough, and we get the low frequencies, and we get the peak. And in cat, the matched voxel is 0.33 cycle per millimeter, which in human refers to 1.5 millimeter voxel. So what did we learn from the cat study? Using myelin-based ferrum particle-based fMRI and size-match voxel sizes, fine-scale irregularities do not contribute to the decoding of orientation stimuli. Why is that? Because the match voxel can only detect very low frequencies. It cannot detect anything in the regime, the peak regime of the irregularity. Now, cerebral blood volume functional MRI that we can do with Mayo is known to show higher functional specificity, fine scale specificity relative to blood oxygenation. Therefore, irregularities also do not contribute to blood oxygenation functional MRI-based decoding. The largest voxel we can expect to capture fine scale irregularities in humans is 1.5 millimeters. Now, Mayo really makes blood vessels, both arteries and veins, transparent to functional MRI. There is so much magnetic properties of the iron inside the vessels that we get no signal from the vessels as well. And because of that, contributions from macroscopic blood vessels and from spatiotemporal propagation of fMRI response are not necessary for decoding oriented stimuli, because we were able to decode without any signal from the blood vessels. Any questions on the Mayo study in cat? I know it's not an easy material, so please go ahead and ask it. Okay. Mechanism underlying decoding orientation stimuli, now with optical imaging, the oxyhemoglobin and total hemoglobin in cat area 18. So we ruled out aliasing. We will test here irregularities, low spatial frequencies, and macroscopic vessels. We use the system of optical imaging, and we image the brains of cats. And remember, we have 616 and 530 nanometer illumination, with which we can get responses of the oxyhemoglobin that represents blood oxygenation functional MRI, and total hemoglobin that represents cerebral blood volume functional MRI. That's the image that we get with optical imaging. Again, in gray is cortex, the exposed cortex, and the black curves are cortical blood vessels, or pial blood vessels on the surface of the brain. Now, we use the machine learning technique to classify blood vessels and gray matter, and we also classify veins and arteries, veins in blue, arteries in red. And again, we computed the contrast to noise ratio. So this is the response, a differential orientation, or vertical orientation against horizontal, based on the oxyhemoglobin, blood oxygenation. So we get responses in area 18, but it doesn't look very sharp, right? The cortical cores do not look very sharp. We also computed the standard deviation, and we got the contrast to noise ratio, which is not really very high. The total hemoglobin, we get sharper orientation columns, and the contrast to noise ratio shows clear patches. Okay? We get responses that are clear for vertical and horizontal orientation. Again, we do the exercise of frequency-specific differential contrast noise and contrast to noise ratio. So this is the responses, the differential contrast to vertical against horizontal grating. This is the standard deviation, and this is the contrast to noise ratio, the mean response divided by the standard deviation. And we find the peak, and we find also the pixel, the size of the frequency that matches pixels in the human visual cortex. We get again a Gaussian response. Note that we don't have here any distinct low frequency responses. They all merge with the Gaussian. It's all based on a Gaussian. So here, unlike in V1 or area 17 of the cat, we can detect responses, we can decode responses that belong to the irregularities. The entire regime is still within the Gaussian around the peak. So it can be considered an irregularity. So the size match voxels in human would be 1.8 millimeter, and we do detect irregularities in cat area 18. So the size match voxel, based on the main frequency of the organization, captures frequencies at the tail of the Gaussian light contrast to noise ratio. Now, how about blood vessels? So here you can see the decoding results. Okay, let me see the scale here. We try to decode one of two orientations, vertical against horizontal. So the chance probability is 50%. Somebody who knows nothing about the experiment can guess whether we presented vertical or horizontal and still be successful at the 50% chance level. So here's the scale bar going from 50% all the way to 100. And the difference is, like here we see the difference, and it is minus 15 plus 15. So let's see first without the difference. Here is the decoding results with data only from gray matter. Remember, we segmented blood vessels, so we mask them out, and we look at the decoding only based on signal from the gray matter. So we can see bluish cyan, meaning on the order of, say, 70% upwards. Large vessel, by the way, whatever is color shows statistically significant decoding above chance level, right? So almost any frequency that we use and any beam size can decode in cat area 18. How about the large vessels? We get lesser extent of decoding rate, successful decoding, but it is still statistically significant. We can decode the orientation based on only responses from blood vessels. And when we combine gray matter and large vessel, again, we get the cyan responses around 70% with statistically significant responses. Now, do blood vessels actually, are they necessary? So here we do the exercise of taking this decoding rate of gray matter and large vessels and subtracting the decoding rate that we obtain from gray matter. We see that nothing remains statistically significant. So this means that, let me tell you first the results of this exercise, where we used again the decoding rate from gray matter and large vessels, and now we subtracted the large vessels. We still get here statistically significant decoding rate, and relatively high. That's the scale here is for the difference. So looking at these two results, we can decode based on large vessels, but the information in large vessels is redundant. We can also detect based only on gray matter without the large vessels contributing any additional information. Please.
What's the difference between the graph you're talking about and the first one that's just gray matter? Like, what's the difference between gray matter and large vessels minus large vessels?
Right. So first, let's look at this. So here is the decoding rate based on only pixels that represented gray matter, okay? And I represented as a function of cutoff frequency, we did low pass frequencies, and beaming. We look at larger and larger voxels. So with gray matter, we can decode pretty well. Large vessel, we can also detect, and we can also decode. It's color. The only place where we don't decode is really when looking at basically DC, no frequencies at all. So we can decode with large vessels. And on the right, it's the combination of information from gray matter and large vessels. We can also decode. Now, what's the difference at the bottom? Here, we took the decoding rate for gray matter and large vessels together, and we subtracted the decoding rate obtained only from gray matter. So here, we get in principle only the information from large vessels, okay? And on the right, it's gray matter and large vessels minus, sorry, on the left, we subtracted the gray matter. On the right, we subtracted the large vessels. And we can still decode, okay? So while large vessels do carry information on the orientation, their contribution is redundant relative to what gray matter brings to the table in terms of decoding. So this is with blood oxygenation, with deoxyhemoglobin responses. Now how about blood volume? I told you that people use blood volume as well. And blood volume is studied now, based on 7 Tesla results in humans, to be a very spatially specific functional MRI signal. It's now the preferred functional MRI signal for high resolution imaging of cortical column. So again, with gray matter, we can decode pretty well. We can also get decoding, statistically significant decoding based on large vessels. And when we combine all the information from gray matter and large vessels, we also get a pretty good decoding rate. Again, here we take the gray matter decoding combined with large vessels and we subtract the decoding rate of gray matter. And we get basically nothing, no statistically significant response. When we subtract the large vessel decoding rate from the combination of gray matter and large vessel, we do still get statistically significant decoding rate. Again, supporting the idea that large vessels can decode information on orientation, but the information they bring to the table is redundant. How long?
5 minutes.
5 minutes, all right. So conclusions from the optical imaging study, irregularities from low frequency tail of the spectrum in the organization of area 18 underlie orientation decoding. While responses from macroscopic vessels are orientation selective, their contribution to decoding are redundant. So overall conclusion about decoding. Multivariate pattern analysis is a powerful technique, but there is no magic. Contributions, what I mean by no magic is that with voxels that have X dimension, right now, they have a certain size, you can only detect signals that you can detect according to the Nyquist frequency with this size of voxel. You cannot detect more than that. Contributions from irregularities depend on the features of the organization, such as the peak frequency, the width of the Gaussian around the peak, and the voxel size. Whether irregularities or large-scale organization contribute to decoding depends on features of the area-specific columnar and low-frequency organization. So in CAT area 17, we show that we cannot get really decoding based on the fine scale on the irregularities. In CAT area 18, where the columns are wider, we did get contributions to decoding based only on the fine scale. While responses from macroscopic vessels can be orientation selective, their contribution to decoding are redundant. So at this point, I aim to go and analyze the spatial specificity in humans, but I think that we need to stop here, right? So I'm happy to take questions.
Yeah, thank you. I think that the main reason we did the seminar before ended because many students have classes.
Yeah, sure.
So we wanted to. So very interesting question. You have for this one.
Please.
How did the virtual binning from your optical imaging of the cat's cerebral cortex, how does that binning compare to the voxel size of an fMRI? Because the dimensions weren't like exactly the same?
Right, so that's a very good question. Look, the reason we use optical imaging is that the signal to noise ratio it gives is amazing. So in principle, each pixel in optical imaging has a size of 17 microns. When we do functional MRI in humans, we can do now at 3 Tesla, 2 millimeter voxels, it's a much larger voxel, and at 7 Tesla, 0.7. So it's much better. We can detect ocular dominance and orientation more or less in humans. Direct, I'm not talking about decoding, I'm talking about direct imaging. Now, remember the matched size voxel that I represented, right? So the beaming that I did eventually, I transformed the result from optical imaging in the cat to what would that mean in humans. And in area 17, the sized matched voxel that was able to get responses for irregularities would be 1.5 millimeter in humans, which means that at 7 Tesla, where we can image now at 0.7 millimeter, we can decode orientation easily. And in cat area 18, it was a little bit larger, 1.8 millimeter, which means that even at 3 Tesla, with 1.8 millimeter voxels, we can decode. So again, optical imaging gives us so much better signal-to-noise ratio. We can go to tiny voxels. Of course, you know, you can do two photon. I'm not sure if you're doing that here. So 2 photon gives even higher resolution than that. But optical imaging is a good tool for us to mimic functional MRI.
Yeah, we have here, for people that are interested in these tumors, they're doing infrared. It's also a very well-known person, spinal visual cortex, is using the multiphoton imaging. Some of you may be in his lab. So I have one question. Clinton has demonstrated this organization of communities of vessels. So whenever you get a penetrating artery, it differentiates to communities that are very well limited in space.
Correct.
How this have an impact on this conclusion?
Well, so the whatever $1 million question is neurovascular coupling. You know, there is like a region, even one cortical column that responds. Which vessels, which arteries eventually delay in order to feed blood to this cortical column? The current concept, as I'm sure you know, is that there is a signal which is transmitted back from the capillaries all the way via arterioles, all the way to the arteries, right? A very specific signal that goes through the arteries back, and that what feeds regions that increase their responses. So that's the question. Yes, cortical blood vessels are organized in communities. There are arteries penetrating from the pia into cortex, and they go directly and they feed capillaries around it. But eventually, which blood vessels get the signal that they need to delay in order to feed even a point-like response? In the part that I didn't present today, I analyzed the spatial point spread function of functional MRI in humans. It's again fitting the model to data, etc., and we were able to show that point function is about 1 millimeter, a full width at half max of the Gaussian response is about 1 millimeter when using gradient echo functional MRI, which is most commonly used, and it's a little bit lower, about 0.8 millimeter, using spin echo functional MRI at 7 Tesla.
Yeah, well, it's the signal that Shmuel was referring to, that is neuron talk to the capillaries, the potassium, it propagates a hyperpolarization back to the penetrating arteries. The whole theory of this was demonstrated by one of our PhD students, published in PNAS a couple of years back. It's work by Mosford, and in collaboration with them, with Cohen, I know Mark. So any question, guys?
Please.
As someone who's not familiar like that heavily with fMRI and cortical columns, I guess from a clinical standpoint, how would you see this redefining kind of imaging paradigms in a clinical setting?
That's a very interesting question. Well, I think that my laboratory now is using, for the first time, decoding multivariate pattern analysis in order to detect the effect of optic nerve neuropathies, such as glaucoma or optic neuritis or trauma to the optic nerve, to detect them with functional MRI. Okay, so we present stimuli and we look at decoding as a function of the region in the visual space. And we expect, we didn't do the experiment yet, we just started, but we expect to be able to demonstrate, to contribute information to the diagnosis of the optic neuropathy and the region that is affected. The region, I mean, in the visual space, which is affected by the optic nerve neuropathies. So it didn't go decoding is used so much in cognitive neuroscience to understand representations in the brain. So the first paper is cited, I looked at this morning, 4,000 times. That's really a lot. So it is really used a lot in trying to understand which stimuli are represented in different parts of cortex. But I believe that my lab is pursuing now the first, we are testing whether we can use it in the clinic. Please.
I'm wondering how you think some of the blood-related measures might relate to non-invasive electrophysiological signals such as electroencephalography, in particular magnetoencephalography, which has recently been shown to be able to decode orientation-level information or column-level information from visual cortex.
Right.
Yeah.
Well, I would be interested. I don't know about MEG showing real cortical responses. I would be interested in that. It would be great to hear about that. But you know, so I do a lot, and I know that Coulter is pursuing similar things, really relating the hemodynamic response to neurophysiology. And you're asking how they relate. So the current concept, and again, this is an evolving theory because these questions are addressed starting in the 80s when people used PET. And okay, how do we get PET cerebral blood flow? What does it represent? So there is a huge debate on whether it represents synaptic activity or action potential activity. And in my view, it's not completely resolved. And in my view, both synaptic activity and action potentials contribute to neurovascular coupling. And at the end, the best resolution you get with neurophysiology, there's no question about that. Functional MRI, MEG, and definitely EEG show like more low frequency responses. But with time, I believe, I think that there's no question that functional MRI has the best spatial specificity, meaning if you show cortical columns or responses to cortical columns, there's no question that functional MRI would do better than MEG and for sure EEG. But whether, to which extent functional MRI and MEG and EEG can represent the real cortical columns is a debate. In CAT, it was shown. So people used, in my lab as well, functional MRI and optical imaging and neurophysiology and showed that the hemodynamic response shows exactly the cortical columns. In humans, everything is more difficult because the magnetic field and the size of the voxels do not allow us to do it in an easy manner. We did demonstrate ocular dominance for humans, and people demonstrated something like orientation, although it's not clear exactly. But it's on the edge. Functional MRI is on the edge. It's not easy to get cortical columns with functional MRI. Please.
I'll make this short. So I hear you're saying optical imaging and you're talking about functional brain activity. So when you say that, you're referring to FNIRS, and if that is the case, like when you're looking at these physiological parameters like blood oxygenation or cerebral blood volume, like do you is there like a consideration for other non-physiological parameters that could also affect? Because you know, I was listening and you were talking about the irregularities and the large scale, and I was trying to figure out what exactly that meant. Was that more physiological parameters, or was that not? So my question is kind of twofold. So, go on.
Well, first, you know, you are mentioning NIRS. NIRS is just the same as the optical imaging that are used in animals. The only difference is that NIRS is used in humans through the skull usually, or yeah, I mean it is through the skull. The system that I presented here, we remove, it's used mainly in animal models, rarely also in neurosurgery of humans, okay? And either in neurosurgery of humans, definitely in animal models, we remove the skull, we remove the pia mater, and we image directly from cortex. But the principle of absorption of light, different absorption by deoxy and oxyhemoglobin, is the same as NIRS. Now, so this is functional physiological. I'm not sure about the second question that you asked.
Also, yes, so functional physiological. I guess when your the results you're getting, was there like a consideration for like non-physiological? Because I saw the irregularities. I was wondering if that was part of physiological parameters, or if that was more non.
So my answer to that would be no. And the reason is that everything that we do with functional MRI and with optical imaging and with NIRS is relative. You have some baseline without a stimulus, then you show a stimulus, and you subtract the signal that you get with the stimulus, you subtract the signal with no stimulus from the signal with the stimulus. Okay, so it's a relative response. It's a percent change response. Whatever is static, non-physiological, is supposed to be washed out.
So that is filtered?
It's filtered out.
Okay.
So philosophically, why the brain visual cortex need to differentiate orientation in a oscillatory?
Oh, that's an excellent question. I'm really enjoying this discussion. So there are many people who do modeling on that, okay? It used to be really a very favorite question of modelers. And there are still people who do that a lot. The main concept, and it was put by Newell and Reason, is that the reason is to minimize wiring length. So we have retinotopy. Retinotopy represents the visual space mapped continuously. It's a continuous transformation from the visual space to cortex. And the reason is that neurons that represent nearby regions in the visual space are connected through axons. So you don't want to do like a telephone directory, you know, each neuron is positioned, neurons are positioned randomly. If you position them in a continuous transformation from the visual space to cortex, then the wiring length will be shorter. Because nearby neurons that represent nearby regions have axons communicating. The same is for orientation. So orientation selective neurons that represent more or less the same region are connected through axons. What they do is they make small responses more with better signal to noise ratio because they communicate collinear representation neurons. Like this neuron represents a small edge here, this neuron represents a small edge here. They communicate and they make representation of longer edges.
So why the tonotopic distribution in the auditory cortex doesn't have that oscillation? If we follow that principle.
Well, okay, that's again an excellent question because the tonotopic is similar to retinotopic. It is space. It is not like, you know, like, although I would be very interested to discuss the auditory. I think that there are features that might be oscillatory and people do not know about it. But tonotopy is a continuous space, okay? It doesn't go, the space, in orientation, you start with -90, it's like a sinusoidal. You start from 0, you go to 90, -90, and go back on. So it is oscillatory. The space, the retinotopy, the visual space, starts on the right side and goes continuously to the left side. And tonotopy as well. You start from 0 frequency and you go to very high frequency.
So we
that's
have,
why.
we have Andy here, which is the expert in auditory. So what do you think?
Sorry?
I think pitch.
Pitch is
oscillatory.
Yeah, so it would be interesting because, you know, I would expect that there might be an oscillatory organization that was not detected yet.
Yeah, for oculars.
Right.
Yeah.
Okay.
On that same realm of philosophical questions, why is it that so much of the brain B1 and B2 of cats, say, is dedicated to this, you know, orientation? Like, it didn't seem like any region in that visual cortex was not dedicated to determining orientation. Like, why is that? Why is it that every single part of the brain in these cortexes, at least one area, or this box layer you're looking at, is dedicated to distinguishing
Orientation.
yeah, like, a better question would be like, why isn't there an area that does not
Oh, there are areas that do not.
within these visual cortex?
Well, there are areas that do not. Say, and let me tell you about the monkey. It's more representative of humans. In B1, it's all about orientation and also retinotopy, right? The visual space. So each neuron represents a small space in the visual space plus a preferred orientation in V1. In V2 is an interesting area because there are stripes that are interleaved. Two of these stripes, different types of stripes. Two of them represent orientation. One of them represents high frequency organization for orientation, and the other low frequency. The two others represent color and luminance, black against white. So that you get like 4 different areas in one area. Then you go to MT, middle temporal. It represents orientation, but mainly visual motion. You present random dot stimuli, MT responds like crazy. And it responds also to orientation, but less so. So why is orientation important? If you guys show here an image processing course in order to understand the image, the very first function that people who do image analysis do, they do derivative. It's a circular derivative, right? It's a gradient in order to detect edges. Edges are so informative on the visual scene. I see you because I see the edges of your shirt, you know, your face, your eyes, all that is represented in edges. Color is kind of giving some additional information. There are many creatures that do not have actually any color perception. But the edges are so informative. So we believe the concept is that orientation selective neurons detect edges in the visual scene. And they send it to so-called higher visual areas, V1 to V2 to V3 to MT, middle temporal, V4, that analyze the edges and try to make sense of them in terms of, okay, what is this in the scene, right? So I see the desk, I see the students, I see the whatever.
Yeah, right. Actually, this decoding that happened, not the same decoding we're talking here, the decoding from the image to more high level concept, need the edges because you first need to know what is there and you need to know where is that you are looking. What object that this goes through 2 different pathways. So the dorsal, I mean
The dorsal is where?
Where, and then the what, right? So these 2 concepts will travel different, by different pathways and will integrate at some level, but those need to go through a filter. So you get, the edges is the basic. To see what the object, you need to know where the limit of the object is, and to see where the object is moving, you need to have that limit decoding at one particular time and the same limit decoding at another particular time, and that will go to before and T. Okay, I think let's give him a very round of
Thank you very much. Thank you very much for listening, for attending, and asking excellent questions.
Okay, so we will continue to remember for the students that will attend the lunch, I think the other student will be in the lunch seminar. And Andy, maybe we have some time that you can talk to him. You have time to 10:30. That will be the next. We want to do that.
So I'm good. I think that now I don't have you for half an hour.
No, that's in the, no, now it was,
Okay.
but, maybe we have some
I'm able to
time.
discuss.
And, we should probably people here. New.
Personally, I have no idea how to solve anything. And then she said also, asked us, oh, I didn't show you anything yet, but it's new in less than 2 weeks.
For the couple of faculty.
Yeah.
Yeah, we should tell her to give us
But you want to meet, you want to.
I just wanted to show
That's Well, you know,
I can send it to you.